I left a board on the bottom on the right that I didn't get through explaining last time. I'm going to save that for the end of today if we have time, and if not, for the beginning of next time where I know we'll have time. So if you're confused about any of those notes, don't worry about that. We'll get there. Today what I want to do is to discuss one of the most active areas of research on part of what Francis Crick, in an act of really unmitigated hubris, named the central dogma of molecular biology. You can say a lot about molecular biologists, but humility is not part of their description. The central dogma, as most dogma proves to be, is not 100% reliable. But unlike most dogma, the central dogma has been pretty good in predictive and explanatory power for most things that happen in the nucleus of cells. So let's just go over what the central dogma is. Now, its cartoon version, of course, is that DNA makes RNA, makes protein. But let's sort of amplify upon that a little bit, because DNA, of course, does other things. DNA is the template for its own replication. We saw that in relative detail a couple weeks ago. The making of RNA is a process called transcription, which is all too often confused with the similar sounding but totally different word translation. Translation being the act of synthesizing a protein from the information in the RNA. So what we're really looking at here is an information transfer process. We're taking information that's stored in this incredibly robust molecule. DNA has been analyzed from mummies that have been dead for thousands of years. We copy that information into a transactional form, the RNA, which is unstable. We'll discuss why that's a good thing. And then we'll take that information and transform it chemically into an entirely different kind of repository of information, making proteins. And of course, you know that proteins come in two types. Some proteins are structural proteins in cells, like actin and the tubulin that we talked about that make up the microtubules. And the other proteins are enzymes. And about half of proteins in a cell are structural, about half of them are enzymes. And for many, many years, it was believed that an enzyme was a protein and that only proteins could be enzymes. What we'll see today is that the central dogma has taken a hit. I'll tell you about the enzyme form of certain RNAs. What I won't have time to tell you about today is other dings to the central dogma, including the ability to take RNA and make DNA from that. And I'll also not be able to tell you, even in this class, about enzymes which make RNA from RNA. We'll save that for MCB 110, so hang on. That's going to be a lot of fun. But today what we're going to concentrate on is this process of transcription by which information that's in DNA is converted to RNA. And there's an enormous amount of mechanistic detail that I'll leave out. But what I want to do is to set us up for next Wednesday's discussion, where we'll talk about how prokaryotes control transcription. It's going to be very, very interesting. And that'll be a prelude to next Friday's discussion about how eukaryotes control transcription. Because the control of transcription, the so-called gene switches, are one of the ways in which cells integrate all of the things that they have to do in an orderly fashion. So what we'll start with is a discussion of transcription in bacteria, and then move on to eukaryotes. And the reason primarily is that because E. coli and its friends were such favorite laboratory pets for many years, it was possible to study common problems to all life in bacteria first, and then once we figured out how to do those experiments, how to make those measurements were taken on into eukaryotic cells. So let's start with a typical prokaryotic transcription event. And typically in prokaryotes, the act of transcription produces a messenger RNA that encodes more than one protein because it encodes more than one gene's worth of information. In prokaryotes, an RNA will usually go through one open reading frame, also known as a gene in the molecular biologist vernacular, and another open reading frame, and maybe up to 10 open reading frames, although in E. coli, the average messenger RNA encodes 2 different open reading frames or 2 different genes. Transcription always starts at a promoter, a singularity at one end of a gene, and proceeds unidirectionally through that gene, making RNA in the same chemical direction as DNA polymerase makes DNA. That is, RNA is always made in a 5' to 3' direction. So here we have messenger RNA being made from the promoter up to a stop sign that is written in DNA, which I'll refer to as the termination sequence, where RNA polymerase will be caused to stop. So that introduces RNA polymerase, which is the enzyme which recognizes promoters and does transcription. So the promoter then is defined as the site that is bound by RNA polymerase and where transcription starts. So what I'll do in here is to blow up our view of the promoter, and we'll recognize that there are 2 parts to a promoter. There is the so-called -10 sequence and the so-called -35 sequence. What the -10 and -35 refers to is the distance in base pairs from the first base of DNA that is copied into RNA. So this is 10 bases upstream, so to speak, of where the RNA actually stops. And this is 35 bases upstream from where the RNA starts. Now, the sequences here are highly characteristic of all genes in the case of the -10 sequence and of classes of genes in the -35 sequence, because it is at the -10 sequence that RNA polymerase nestles down onto a gene and binds to a gene for the very first time. The -35 sequence is recognized by a protein, a family of proteins referred to as sigma factors, and these are regulatory proteins that help RNA polymerase know what genes to transcribe at what time. So let's drill in a little deeper on this. Now, RNA polymerase of E. coli was actually discovered by Mike Chamberlain, a Berkeley professor who was here for many years, just recently retired. And this is one of the great oversights of the Swedish Academy not to have given Mike a Nobel Prize for this. Bacterial RNA polymerase is made of 4 different subunits, 2 copies, or 4 subunits, 2 copies of the big subunit called alpha, 1 copy each of beta and beta prime. Typically, multi-subunit proteins are referred to in Greek nomenclature in a descending form from the largest. So alpha is the largest, beta is the second, and beta prime is the third largest subunit. These 4 polypeptides make up the core enzyme, often called the holoenzyme, in case you come across that word. I don't care if you know it or not. And then the sigma factor, as I said, is the regulatory subunit that comes in many varieties. So most genes that you would call housekeeping genes, genes that have to be on so that a cell can live, are transcribed by a sigma factor referred to as sigma 70 after its molecular weight. Biochemists tend not to be very creative in the naming of proteins. They name proteins after their molecular weight. There are other sigma factors, like the sigma factor that I'll not bother you with the size of. I'll call it the sigma for heat shock. Heat shock is a kind of stress that bacteria exhibit. In order to deal with that stress, they have to turn on a bunch of different genes, and so there is a sigma factor dedicated to heat shock. And that sigma factor will recognize a particular sequence at the -35 position of genes that have to be turned on at heat shock. For bacteria, unlike E. coli, that go through sporulation, a developmental program in bacteria, there are sigma factors dedicated to sporulation. So there's quite a large number of sigma factors, and I think new ones are still being discovered. The key thing about sigma factors is they provide specificity to particular promoters by recognizing variants of the -35 sequence. RNA polymerase always recognizes the -10 sequence, but for RNA polymerase to engage in transcription, it depends upon the help of a sigma factor. Any questions so far? You'll see that the theme of sigma factors gets carried over into eukaryotic regulation, but amplified in elaborate ways. Yes? They're just sequences at minus 35 position or minus 10 position in nucleotides from where transcription starts. And those are the landing sites for RNA polymerase at minus 10 and for sigma factors at minus 35. Any other questions? Okay. So let's go over what makes RNA special. And the thing that makes RNA special is, in fact, this particular hydroxyl, the 2' hydroxyl on the sugar ribose. You'll recall that DNA is made of deoxyribose sugar, which lacks that hydroxyl. And this hydroxyl is what makes RNA unstable, vulnerable to attack by enzymes or base or a variety of different kinds of biophysical conditions. So RNA is designed to be unstable rather. So why do you think we would want RNA to be unstable? I think Jennifer mentioned, and as did I, that it is believed now, and I think it's almost certainly true, that the earliest forms of life used RNA as its genetic material. So isn't it curious that RNA is fundamentally unstable? Why would we want to use an unstable macromolecule to involve in the control of gene expression? Why would you want to do that? Yeah, what do you think, Jared? Yeah, Jared's got it exactly right. We would have molecular cacophony if RNA were stable. It would be like holding down the hold pedal on a piano as you play. If you can't let the signal attenuate, you would have a mess. So that's a really, really important part about RNA. The other important part about RNA is that it uses the base uracil instead of thymine. And the other part about RNA synthesis that's really important is that no primer is required for RNA polymerase to begin to engage the synthesis of RNA. That's one of the pieces of evidence that makes us think that RNA was in fact the original source of genetic material in the world, because no RNA polymerase needs a primer, all DNA polymerases do. And as I said before, RNA is synthesized in a 5' to 3' direction by chemistry that's virtually identical to the chemistry involved in DNA polymerization. So I'll not go through those details. Your book does a great job of that. So in discussing transcription, what I want to do is to break it down into 3 phases: initiation, elongation, and termination. And we'll turn our attention to initiation first. So step 1 is the formation of what is referred to as the closed promoter complex. Closed meaning that the DNA double helix is still base paired to itself. So here at the minus 10 sequence, RNA polymerase recognizes the bases there, the base pairs, and binds. And at minus 35, the sigma factor has bound. The next step is the formation of an open promoter complex, which is very similar to something that you've seen before, which is the melting of the duplex at the replication origins. In this case, it's not a replication origin, it's just the promoter, and the interaction between sigma and RNA polymerase results in a melting of the helix over a region that's about 17 base pairs. Now, as an aside, if you're thinking that it's important that you remember 17 base pairs, you're wrong, right? So why do I give you that detail? It's to help you develop an intuition for scale, which is really important for thinking about biology. So let's think about, what does 17 base pairs mean on a helix? In the DNA double helix, how big of a region would that be that is melted open by RNA polymerase? What do you think? Small? Give me some units that would give us an intuition to that. It is a small region. 17 bases is pretty small compared to the 4 million bases of an E. coli chromosome. How many turns of the helix? How many base pairs per turn? Remember? 10 base pairs per turn. So it's almost 2 turns of the helix. So this is a pretty big distortion. And of course, we'll put some stress on that gene that will have to be relieved by other enzymes. And you'll come to those enzymes in MCB 110. But for now, I just want you to get a sense of scale about what's really happening here. Once the open promoter complex is made, RNA polymerase, without need of a primer, can start to synthesize RNA. Okay. Now, I've just told you something rather remarkable, that RNA polymerase binds this particular sequence, sigma binds that particular sequence, and something that you should always be wondering is, well, is that right? How do they know that? How do I know they got it right? And one of the characteristics of science is that we have to be able to tell you why we believe something, what the experimental evidence is. So we'll spend some time talking about a methodology that's been used for years now, although there's some newer methods that are starting to displace it. This is still widely used, to figure out where a particular protein recognizes DNA. Now, proteins that interact with DNA can interact in one of two ways, either at specific sites or non-specific sites. Most of the interesting action with respect to the control of gene expression involves proteins that bind at specific sites. So let's think about how you might identify exactly where a protein binds. And we'll use a method called DNA footprinting. Now, for DNA footprinting, what we need is a piece of DNA that, well, what we need is many, many, many pieces of the same sequence of DNA. How would we get that? Well, we could make that by PCR, or we could clone a piece of genome that, for example, might contain a promoter. So what I'm representing here is many pieces of the same DNA sequence to which we've added a radioactive phosphate, P32, at one end on one strand. Okay? Here is the same piece of DNA, radiolabeled at one end on one strand. So here's what we do. We use what's called a limited digestion with an enzyme called DNase. DNase, of course, degrades DNA by making nicks in one of the two strands. So what I'm showing here is not what happens. What I'm showing here is a DNA molecule that's been nicked here, 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 here. What I wish I could represent is a population of molecules in which each molecule is nicked at a different place. So in aggregate, we have molecules that are nicked at all of these places. Is that point clear? That's actually a crucial point for the experiment to work. This represents, I don't know, an attomole of the same DNA molecule, each being nicked in a different place. Is that clear? Yes. Yes, each DNA molecule in the population gets nicked at a different place. And we use enough molecules and enough DNA, so on average, we can count on some molecule being nicked here, some molecule there, all possible places. Got it? Great, thanks. So what happens? Well, if we add our site-specific DNA binding protein, like a sigma protein, it will bind to a particular sequence. By binding to that particular sequence, it occludes the phosphodiester bond from accessibility to the DNase. So we'll be nicking here, nicking there, there, everywhere, except where this protein has protected it. So now, what we do is to take the products of that reaction and we separate them on a polyacrylamide gel, just a sieving matrix, use an electrical field to separate the molecules, and in a polyacrylamide gel, of course, the molecules separate according to size. So these are the small fragments, those that got nicked near the P32, and these are the big fragments that got nicked far from where the P32 label is. Once we have separated these molecules on the gel, We'll put a big piece of film on top of the gel in a darkroom and expose it, and then develop the film, and the film will have radioactivity in a ladder in the case of the control lane, so we'll be able to see the distribution of molecules, each of which have been nicked one place because of the p32 radioactivity at its end. And in the experiment, what we'll be able to see are these molecules nicked ding, 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 ding here. There'll be a gap where no molecules got nicked, and there'll be big ones representing the dings down here. And it's this gap in the ladder that is the footprint, and because we know exact positions of each of the molecules in this ladder from this end, we can actually write out the sequence that that protein binds. Okay? This is a really cool method. It's been done probably thousands of times by lots and lots of different people. Any questions about that? It's a real versatile method. Could you imagine doing an RNA footprinting experiment? Suppose you wanted to figure out where in an RNA molecule an RNA binding protein bound. Could you figure out how to do that with this kind of methodology? Yeah, it's the same kind of thing. Same kind of idea. Okay? Okay, so that's how we know where proteins bind. We've initiated transcription, and now let's elongate transcription. Elongate, yes, question. That's a great question. Would it make any difference if RNA were single-stranded whereas DNA is double-stranded? Well, the differences would have to be that we couldn't use DNase because that's DNase, so we'd use an enzyme called RNase. But note that I have label at only one strand at one end. That's actually critical. You should think about what a mess this experiment would be if we had label at both ends. And so I'm only looking at one strand anyway, so RNA would not create any problems. Okay? But do think about why we want the label at one end and not at the other end on the other strand. Yes? Pardon me, I didn't catch it. Oh, the question is, would I need a different kind of protein to bind to RNA? Make sure you understand what the goal of the experiment is. The goal of the experiment is to take a protein that you think binds to DNA or to RNA and figure out exactly where it binds. So you would only do an RNA footprinting experiment on a protein that you had reason to believe was involved in binding RNA. Okay? So let's think about elongation. So elongation is nothing more than making an ever longer RNA molecule, with, of course, this being the 5' end of the RNA. So here we are. Our bubble is moving down the DNA. We started transcription up here. These sequences have been copied into a single strand of RNA. The energy for elongation comes from the hydrolysis of the pyrophosphate released after every nucleotide is added, just like DNA. But unlike DNA, we are limping along, relatively speaking, at 60 nucleotides a second. Okay? So how many turns around the helix per second is RNA polymerase doing? 6, yeah. So I say limping, but that's not so bad. What can you do 6 times in a second? Can you make your hand go around in a circle 6 times in a second? Let's try it. Let's always try it. 6 times in a second. Ready? Jeez, didn't make it. Go. RNA polymerase is still moving pretty well, but it's a piker compared to DNA polymerase, which is going at 1,000 nucleotides a second. Why is RNA polymerase going slower? There's at least 2 reasons from what I've told you so far that you should be able to deduce why RNA polymerase is slower than DNA polymerase. Think about that. Any ideas? Yeah, what do you think? What's your name? Sunitha? RNA is not as stable. That's true, but that wouldn't have a profound effect on the speed at which the RNA polymerase could go. But it was a good try, and I did mention that. The answer is a little more subtle than that. You want to try again? Anybody else want to take a crack at it? What precedes DNA polymerase down the duplex to help it out? Have you reviewed those notes? A helicase, right? There's an enzyme in front of DNA polymerase that is burning ATP to unwind the strands. RNA polymerase, as best we know, has no helicase to help it. And in fact, because it's melting the DNA, it's actually putting some twists and some torsion in the DNA that it's got to overcome. That's part of it. What else is going on here? Is there any baggage behind RNA polymerase that might be slowing it down? Right? It's making an RNA longer and longer and longer, that as the text illustrates beautifully, and I've drawn crudely here, every time it goes around, it's got this ever longer and longer rope behind it that has to spin around with it. And, your typical gene is about 1,500 base pairs long. So a transcript is going to be about that long. A polycistronic message, if it has 10 genes, will make an RNA that's 10,000 base pairs long. And RNA polymerase is, along like that. It's actually pretty amazing. And it makes you wonder whether it really works like that. I'll come back to that point a little bit later, but I want to give you an intuition for transcriptional elongation. Yeah, Jared. Because what I'm saying, so the question is, why does DNA polymerase not have the same problem? Because if DNA polymerase were replicating, it's leaving its bases paired with the strand, right? So it's not carrying anything with it. It's just chunking that extra base in and leaving it behind. Good question. Thank you. Everybody got that point? Okay. So far, there's nothing particularly sophisticated here if you understand DNA replication. When it comes to termination, though, it's a little odd, and you will note that I called that thing a termination sequence, and I did not call it terminator. Here's why. The real terminator is much more interesting than that. Here's the termination sequence. RNA polymerase has been transcribing its way along. It sees the termination sequence and is blind to it. It continues to transcribe. Okay? But now, the RNA copy of the termination sequence exists, and there's a short little palindrome in the RNA that was encoded by the DNA. Palindromes meaning the same sequence read this way as that way, which will then base pair with itself to make this double-stranded RNA region here with a loop in between it. The combination of the structure and sequence of this feature in the RNA is recognized by a protein called Rho protein, which recognizes this part in the RNA and causes the RNA polymerase that's already gone by that to stop. Okay? Now, we have a pretty good physical understanding of how that process works because James Berger here at UCB and one of his postdocs, Emmanuel Skordelakis, crystallized the Rho protein and determined its three-dimensional structure and now have some really good mechanistic insights as to how Rho protein stops RNA polymerase. I'll spare you those details for now, but the key thing here is that transcription goes past the termination sequence. The terminator is made in the RNA, and it's an interaction with the RNA and a terminator protein that actually causes DNA polymerase to stop. you might wonder, why not just put a big protein on the termination sequence? I wonder too. This is how it works. Okay? Any questions on that? Yes, sir. So the question is, if rho protein stops transcription, then how do you make rho protein? Okay, so let's think this through. We have on the order of 4,000 different proteins in E. coli. On the average, each transcript encodes 2 different proteins. Okay? So there is a transcript that encodes rho protein that, when translated, as we'll discuss on Monday, makes rho protein, lots of rho protein. And rho protein will recognize a sequence and structure in RNA in its own transcript, as well as all other transcripts, all being in quotes here, and cause transcription termination after the message has been made. This message here will be translated into whatever protein is encoded there, whether that's rho or anything else. Does that help? Okay, good, thanks. Other questions? Yes? Does the terminator sequence correspond to the termination sequence? Yes, the termination sequence is the part of the DNA duplex that encodes the RNA that folds into the structure. Okay? All RNA sequence and structure, except with one exception, and I'll come to 2 exceptions, I'll come to those, come in the DNA. Okay? So that, in a nutshell, is how bacteria make their transcripts. On Wednesday, we'll talk about the regulation of transcription from a particular promoter, so you'll know how that works in quite great detail. We'll move now to eukaryotes, and eukaryotes have 3 classically defined different RNA polymerases, though I should warn you that I'm painting with a broad brush here, and I am ignoring a very interesting class of RNA polymerases that make RNA from RNA. Those are not subjects for this class. So the 3 classes of RNA polymerases are called creatively RNA polymerase 1, 2, and 3. And they have dedicated jobs. In eukaryotes, all messenger RNAs are made by RNA polymerase 2. All ribosomal RNA, the RNA that is actually the structural framework and the catalytic center of ribosomes, we'll come to that next Monday, is by RNA polymerase 1. And the tRNAs are transcribed by RNA polymerase 3. Now, as I've told you, there's an ever-growing collection of new so-called non-coding RNAs. There's a class called the microRNAs that we'll talk about, I think, next Friday. They're made by RNA polymerase 2. And my favorites among this whole class, there's the snRNAs, these small nuclear RNAs that are transcribed by RNA polymerase 2 and 3. And my favorites are the so-called siRNAs, these small interfering RNAs that will do wild things to gene expression next Friday. These were discovered by Andy Fire, a former Berkeley undergraduate who got a Nobel Prize recently for his discovery of these small interfering RNAs and what they do. And there's going to be more. Be happy you're taking the class this year and not next year, because there's going to be more RNAs next year, trust me. Now, eukaryotic RNA polymerases are gigantic enzymes. They are made of 12 to 13 different subunits and interact with hundreds of other proteins at the promoter. And I'll tell you probably more than you want to know about all of those next Friday. Some of the RNA polymerases share subunits with each other, and others are specific to different RNA polymerases. And one of the world experts on RNA polymerase is none other than our own Robert Tijan here at UC Berkeley, who's an expert both on RNA polymerases and eukaryotic gene expression. And so far, the guys in Sweden have not recognized him adequately. Hopefully that will change. I'll have more to say about Tijan's stuff next Friday. So let's talk about making ribosomal RNA first, because that's a relatively simple thing, but really, really interesting. You and I and all eukaryotes, yeast, whatever, have many, many, many copies of the genes that encode the RNA in ribosomes, the so-called ribosomal RNA genes, and we have hundreds of them, usually arranged in a tandem array on one or more chromosomes, depending upon the species. Why do we have so many? Well, you need a lot of ribosomes. Each of the ribosomes have to have its own complement of RNAs. We'll talk about the details of that on Monday. And apparently, it's just impossible to make enough ribosomal RNA from one copy of the ribosomal RNA genes. There's no creature that has yet been discovered, and at this point I'd be willing to say there will be none, that has one copy of the ribosomal RNA genes. Even E. coli have multiple copies of ribosomal RNA genes. So here's what's interesting. The genes are all transcribed together into a transcript that's rather large, about 9 kilobases, okay? 9,000 base pairs long. Now, we need to make 3 different RNAs for ribosomes, something called the 5.8S, the 18S, and the 28S. You'll see what they do on next Monday. And the way we do that is to make a very large precursor and then process that precursor in rather complex processing steps to give us the mature ribosomal RNAs. But here's something very, very interesting. Depending upon the organism and the rate at which the cells are growing, some of these repeated genes are transcribed, and some of the genes in that same array are not transcribed. So isn't that interesting? The sequence of this is absolutely identical to the sequence of that. There might be 50 or 100 different RNA polymerases actively engaged in transcribing this set of genes at the same time, and none transcribing this one. Isn't that curious? There is nothing that I have told you that would let you understand how that can happen. How can we have 50 RNA polymerases transcribing this gene and none transcribing that gene? In fact, there's nothing I could tell you that would illuminate that. This is a really interesting and deep mystery and has to do with the impact of something called chromatin that we'll come to next Friday that helps make this gene different from this gene in some way still not understood. So that's RNA polymerase. It's dedicated, RNA polymerase 1, dedicated to making simply the RNA that we need to assemble ribosomes. Eukaryotic messenger RNAs, as I said, are made by RNA polymerase 2, and their primary transcript is changed in 2 ways. Typically, there is a specialized chemical structure at the 5' end of the message called a cap, and what the 5' cap is the addition of ribo-GTP to the first base in the message that's transcribed in a 5' to 5' phosphodiester bond. So usually when we're making DNA or RNA, it's like 5' to 3', 5' to 3'. In the case of the cap, this extra base is added on backwards. It's there to help stabilize the message against ribonucleases, which otherwise would make the message even more stable than it already is. So specialized caps at the 5' ends of eukaryotic messages. There's this arms race, of course, between cells and viruses, and one of the tricks that poliovirus has up its sleeve is a decapping enzyme. So that when poliovirus infects cells, one of the poliovirus genes makes an enzyme which removes the caps from the host messenger RNA so that your messenger RNAs get degraded and the poliovirus's messenger RNAs do not. Kind of wild. The other thing that happens at the 3' end of a message is the addition of A for about 150 to 200 A's. We call that poly-A. poly A is added by an enzyme called poly A polymerase, which is a stupid enzyme. That's unfair, actually. Maybe it's an intelligent enzyme. It needs no template to add these A's. These A's are not untemplated A's, and they're just a long string of them added to the 3' end for reasons that are going to be interesting on Monday, but the brief answer is that it contributes to the stability of the message. Now, one of the really interesting things about eukaryotic transcription is despite how many people have worked on it, including the brilliant Tej, we still do not have a very good idea about what stops RNA polymerase. We do know how the 3' end of a message gets made, and the way that happens is that the RNA polymerase transcribes past this sequence in the DNA, which produces this sequence in the RNA, and there is a processing enzyme that cleaves the RNA right after this, and then poly A polymerase adds it on. But does RNA polymerase actually know that its message has been processed? I don't know. I mean, it's a really interesting problem. We do know that there are genes in humans, for example, that are bigger than the entire chromosome in other organisms, other eukaryotes. So RNA polymerase can, in fact, transcribe far enough to transcribe an entire chromosome, right? So this is a deep mystery. We do not know what stops RNA polymerase, but what we do know is that most messages are processed from larger precursors at the 3' end. Any questions about that? I'm going to skip RNA polymerase 3. The themes are similar, the promoters are simpler, not very complicated. We might come back to it later. Instead, I want to tell you about a sleepless night that I had in 1977, when I read a series of papers that shattered my ideas about molecular biology. One of our colleagues here at Berkeley, Gunther Stent, published a paper in a book actually, in 1964 called Coming of the Golden Age, in which he declared that all the important discoveries in molecular biology had already been made and that we should all go work on figuring out the brain. Okay? Here is one of about 100 incredible discoveries made since Gunther published that book. Few people have been more wrong in one publication than Gunther managed to be in his. So what we'll use is a method called DNA-RNA hybridization and the electron microscope to make a stunning discovery. Let me set up the method here. If we were working with a typical prokaryotic gene and its message, and this were the DNA and this were the RNA, if we were to melt those at high temperature and allow those to reanneal, the base pairing between the RNA and the DNA is as good as the base pairing between the DNA and the DNA, and we can cook the conditions such that RNA-DNA hybrids are actually more stable than DNA-DNA hybrids. And what we would see if we looked at it in the microscope, the electron microscope, would be something like this, though without color, because electron microscopes are not in color. We would see an RNA-DNA hybrid with what's called a displacement D loop. Okay? So this is the sequence that RNA polymerase originally copied into RNA. This is the non-coding strand. The RNA hybridizes to the DNA and displaces that strand. So, in 1977, two different groups, Suzanne Berger with Phil Sharp at MIT and Rich Roberts and his colleagues at Cold Spring Harbor, essentially simultaneously did the following kind of experiment. Using adenovirus, a virus that infects our cells, it's a respiratory virus, they hybridized its messenger RNA to the adenovirus genome and looked in the electron microscope and saw this. Holy smoke, what in the hell is that? So what we have here is an RNA molecule that is continuous, but not continuous with the DNA that it was copied from. Now, that is bizarre. And the title of the papers in that issue of that journal was The Amazing Structure of the Eukaryotic This or That. Man, I was up all night trying to figure out how this could possibly be true or why it had to be wrong. So how could this be true? How could you have an RNA copy a gene, but only in the mature RNA only have part of the gene represented? How could that be true? How could RNA polymerase do that? There's at least two models. Can you think of one of them? There's a smart one and a dumb one. The dumb one is the truth. Yes, what do you think, Katie? That's a question instead of a model? Yeah, the RNA does not have all of the DNA represented in it, and in particular, it's this part of the DNA that is not represented in the mature RNA. That's why it's looped out in this RNA-DNA hybrid. Any ideas how this could happen? What do you think? So Katie suggests that this is in fact not the first RNA that was made, that something much bigger was made and pieces were snipped out. That would really be a dumb and wasteful way to transcribe our genes because we're burning all of those ATPs, right? About 2 o'clock in the morning in that June 1977, I had a much better idea. I realized that that would be wasteful and that what RNA polymerase must do is to transcribe its way down some genes and recognize a signal that would cause it to jump over part of the DNA and transcribe its way and then jump again, right? That would have been smart. In fact, that's not what happens at all. Katie's exactly right. The whole damn thing gets turned into RNA, and most of that RNA gets processed out, thrown away. Okay? So this discovery of the discontinuous forms of eukaryotic genes earned a Nobel Prize for Phil Sharp and Rich Roberts, a really well-deserved one for this extraordinary discovery. So how does it happen? Well, there's yet another Nobel Prize in the mix. Exons is the portion of eukaryotic genes whose sequences appear in the mature RNA, and as Katie predicted, introns exist, which are the portions of eukaryotic gene that is transcribed but does not appear in the messenger RNA. Why? Because there's a process called RNA splicing that comes in two forms. One form was discovered by Tom Check, a former Berkeley student who got his degree in this building. He did not do the work here at Berkeley, he did the work at the University of Colorado, who discovered that the RNA of the ribosome of tetrahymena, the ribosomal RNA, is in fact an enzyme. And what it does, it splices its own intron out. So here's the transcript of the ribosomal RNA as it's made. It has an intron in it, and Check discovered that the RNA itself is the catalyst for processing out the intron and stitching the primary message, the primary transcript back together again. The intron is looped out in what's called a lariat form with an exposed 3' hydroxyl, and there's some really interesting chemistry that goes on as RNA attacks itself to cut the RNA and to ligate the RNA back together. However, most genes have their introns removed by splicing factors, which use RNA and proteins to recognize the junctions of exons and introns, and then these RNA splicing factors assemble into some big thing called the spliceosome, about the size of a ribosome, which processes the introns out. But wait, there's more. It turns out that eukaryotic genes, and this will take 40 seconds, have typically many exons and many introns. You can splice all of the introns out in order, but there is alternative splicing possible in which you splice the first exon to the third exon, skipping this exon and those two introns. You could splice the first one to the fourth one. And in fact, differential splicing can produce many different polypeptides from the same gene. The record so far is a Drosophila gene known as DSCAM, in which alternative splicing produces at least 23,000 different proteins from the same gene. So that is wild. And what I want to leave you guys thinking about is the transcription of a human gene 1 million base pairs long, recognizing that all of this stuff happens as the RNA is being transcribed. So the transcript has a splicing factory associated with it, doing all of this processing of the exons, and try to wrap your mind away how that might happen inside a cell. So with that, have a good weekend. I'll see you guys either office hours today at 3 or on Monday morning.